Hello all. Today we'll solve question number 40 of GATE Biotechnology 2018 paper. So the question is 5' capping of mRNA transcripts in eukaryotes involves the following events. P. Addition of GMP on the 5' end. Q. Removal of gamma phosphate of the triphosphate of the first base at the 5' end. R. 5 to 5' linkage between GMP and the first base at 5' end. S. Addition of methyl group to N7 position of guanine. So, which of the following is the correct sequence of events? Option A, P, Q, R, and S. Option B, P, R, Q, and S. Option C, Q, P, R, S. Option D, Q, P, S, and R. So to understand the steps involved in one of the processes of transcription, we learn everything about transcription, which is one of the major biological processes. So cellular processes in which RNA is synthesized using DNA as a template is known as transcription. In this video, we'll look into how RNA is formed during transcription and also look into what happens to the RNA after transcription. So before that, what is RNA? So RNA is a polymer of ribonucleotides held together by 3' to 5' phosphodiester bridge and are single-stranded as compared to the DNA, which has 2 strands, which is double-stranded. So RNA is the only molecule known to function both in the storage and transmission of genetic information and in catalysis. So it has the genetic information of the DNA, and also it sends information of how the proteins are formed. So all RNAs except the RNA genomes of certain viruses derived from information which is stored permanently in DNA. There are several types of RNA molecules that can be classified based on their size and function. There are 3 major classes of RNAs. So one of them is mRNA or mature RNA. It constitutes 5 to 10% of all the RNA. So these are basically transcripts from which proteins are synthesized. So they are the protein coding transcripts. The other class is tRNA or transfer RNA, which account for 10 to 20% of the RNAs. So these tRNAs read the mRNA sequence and transfers appropriate amino acid to the mRNA during protein synthesis. So this is a crucial RNA molecule that's involved in the protein synthesis process. So another such RNA molecule is rRNA or the ribosomal RNA, which constitutes the majority of RNA, like 60 to 80%. So these are constituents of the ribosome, and they are also involved in protein synthesis. So there are many additional specialized RNAs which have catalytic activity or regulatory functions, and all these are also present in the cell. So some of them are microRNAs or miRNA, and ribozymes or U1 RNAs or long non-coding RNAs, and so on. The process of replication happens for the entire DNA molecule, whereas the process of transcription is highly selective. This selectivity is due to signals embedded in the nucleotide sequence of the DNA that instruct the enzymes of where to start and where to stop and how often to start the process of transcription. So many of the RNA transcripts are synthesized as precursor molecules, and these are known as primary transcripts. So these, upon further modifications and trimming and several other processing steps, these primary transcripts are converted to functional RNA called the mRNAs or mature RNAs, which code for proteins. So the transcription of all RNAs in prokaryotes takes place in the cytoplasm. Prokaryotes are organisms like bacteria and archaea that do not have a nucleus and several other organelles in the cell. So most of them are unicellular. While in eukaryotes, that is the higher order organisms like animal cells and the fungi, the process of transcription takes place in the nucleus and in the mitochondria. So mRNAs are synthesized in the nucleoplasm, which is a component of the nucleus excluding the nucleolus. The basic biochemistry of RNA synthesis is similar in both the prokaryotes and the eukaryotes, but its regulation and the functions are more complex in the eukaryotes. So we'll look about the overview of transcription in both the prokaryotes and eukaryotes in this video. So RNA synthesis in prokaryotes is catalyzed by a large enzyme called the DNA-dependent RNA polymerase. So this RNA polymerase synthesizes all types of cellular RNAs in prokaryotes. It uses one of the DNA strand as a template on which the complementary ribonucleotides are incorporated to synthesize the RNA. For example, in this sequence, this strand of the DNA is that acts as a template strand, and it produces the RNA. So here we could see that the sequence of the RNA is complementary to the basis of this template strand. So C, G, and in place of thymine, T nucleotide, uh, the RNA molecule has a uracil. So the complementary base of A is T, but in the RNA we have U. So here G, C, the complementary of T is A, and so on. So the RNA polymerase enzyme synthesizes RNA in the 5 to 3' direction. That is, the DNA template is read backwards in the 3 to 5' direction. So in the RNA, like I said, thymine is replaced by uracil. So moving forward, the prokaryotic RNA polymerase, abbreviated as RNA-pol, is a multimeric enzyme. It consists of 6 subunits. So these 6 subunits comprise of 2 identical alpha subunits, similar but not identical beta subunits, so hence they're called beta and beta prime, and an omega subunit, and another factor called the sigma factor. These 5 subunits together form the core enzyme, and the core enzyme along with the sigma factor is called the holoenzyme. So these are the sigma factor is essential for the core enzyme to bind with the DNA during the process, to initiate the process of transcription. So a single RNA polymerase enzyme performs multiple functions in the process of transcription. So it searches for and it binds to the promoter site in the DNA sequence. It unwinds a short stretch of the double helical DNA, and then it selects correct ribonucleotide and catalyzes the formation of phosphodiester bonds. That is the polymerization according to the base pair rule. So it forms bonds between A and T, G and C, and likewise. And then it also detects the termination signals, that is like when to stop the process of transcription along the DNA sequence. It also interacts with activator and repressor proteins that regulate the rate of transcription. Transcription in prokaryotes consists of 3 stages. So first is the initiation phase, where the RNA polymerase enzyme recognizes the promoter in the DNA and starts the transcription. Next is the elongation phase, where the RNA strand is continuously growing and as it is being synthesized. So the final one is the termination phase, where the RNA polymerase stops the synthesis when it reaches the stop site in the DNA, and the nascent RNA is separated from the DNA template. So, like we just saw, the initiation of transcription involves the interaction of the RNA polymerase enzyme with the DNA at a specific site called the promoter. So, this promoter is usually located on the coding strand. So, simple promoters are found in prokaryotes. So, in the promoter region, 2 general types of sequence elements are found. So, one sequence element is believed to promote initial binding of the RNA polymerase enzyme. So the other element is usually has high content of A and T bases, and they are 6 to 8 nucleotides in length, and these two sequences are located about 35 and 10 bases upstream, that is before the start site of the transcription. So change in just one pair of this promoter region decreases the rate of the transcription. So this is a simplified representation of the initiation step of transcription. So this is the RNA polymerase enzyme. So the core enzyme, that is the 5 subunits of the RNA polymerase, along with the sigma factor, so these together constitute the holoenzyme. So these together, they help, they recognize the promoter region in the DNA, and they bind to the helical double-stranded DNA structure. So the enzyme then helps unwind the double helical strand of the DNA sequence. So then the sigma factor is released from the enzyme, and then the core enzyme carries out the next two steps. The next step in transcription is elongation. So now the double-stranded DNA is unwound, and as the RNA polymerase enzyme moves along the DNA, complementary base, it adds complementary bases to the existing, the template DNA strand, and the RNA molecule keeps growing in length. So yeah, elongation is, as the name suggests, it's just the elongation of the RNA molecule. So this is the nascent or the primary transcript that is being formed. So here's a detailed representation, like a zoomed in view of what happens during transcription. So this is the RNA polymerase enzyme. It binds to the double helical DNA, and as the RNA polymerase moves forward, it unwinds the strands, and yeah, behind it, the DNA molecule again, it rewinds to form the double helix structure. And yeah, so here's the template strand and the other non-template, which is the coding strand, and this is the new RNA molecule that is being formed. So the final step is termination. So this is where the transcribed region of DNA template contains the stop signals, and the enzyme recognizes this stop signal, and the whole process of transcription stops. So prokaryotes have two classes of these termination signals. So one of the termination signals relies on protein factors called Rho, the Rho protein. So this process of termination is called Rho dependent termination, and the other one doesn't rely on a Rho factor, and hence it's called Rho independent termination. So let's look at the first type of termination called the Rho dependent termination. So this is signaled by a sequence in the template strand of the DNA molecule called the transcription stop signal. So this sequence is 40 bases in length, and it constitutes inverted repeats. So we'll just look at what this means in the next slide. So these inverted repeats are recognized by another protein called a termination protein or the Rho factor. So Rho, the Rho protein, is an ATP dependent, that is, it depends on ATP, and it's an RNA stimulated helicase which binds to the signals. So once this protein binds to the signals, the unwinding of the DNA helical structure does not happen. So the RNA polymerase cannot move any further because the DNA molecules are still bound together. So because of this, the RNA polymerase dissociates, that is, it gets disconnected from the DNA, and this disrupts the nascent, the new DNA and the RNA complex. So it breaks this complex, and hence the new RNA molecule that is formed is released from the DNA template. So this is how Rho dependent termination works. So the other type of termination is Rho independent termination. Most Rho independent terminators have two distinguishing sequence features. One is palindromic GC region, which is followed by a region that is rich in A and T. So thus the RNA transcript of this palindrome is self-complementary sequences, permitting the formation of a hairpin structure. So what is a palindrome? A sequence that is inverted and it is read backwards. It's basically the same thing as you'd get when you read it forward. For example, just take the word swims. It has SWIMS. Now when you just invert it, like put the letters upside down and read it backwards, you still get SWIMS. So this is what you call a palindrome. And yeah, so when you just, so these two sequences, they are complementary to each other. So when we see such pattern in RNA, like we see a stretch of G's and then the complementary to it is C. So we have a stretch of G's and then we have a stretch of C's. So this could fold and they could complement with each other, and this could result in the formation of a hairpin structure. So that is what the first sequence feature is. So the second feature is a highly conserved string of A residues in the template strand that are transcribed into U residues. The RNA transcript ends within or just after them. So to have a better understanding, this is what the transcription termination site looks like. So let's say these are the sequence signals. So these are like palindromes. So for example, like when you read this sequence backwards, you get this sequence. So this is a palindrome. So whereas inverted repeats, so which are the sequences for row dependent transcription termination. So inverted repeats just means that you have like CCC, TAGC, whatever, and then again you have CCC. So that is what you call inverted repeats. So yeah, so this is like the sequence structure in the row independent transcription. So what happens is, since this sequence is complementary to this sequence, in the new RNA molecule that is formed, this binds with this and it forms a hairpin like structure. So after this, the transcription cannot happen further, and hence it just causes the RNA nascent molecule to be released from the DNA template. So this is the row independent transcription. It doesn't require any external row proteins to bind to the DNA to stop the transcription. So yeah, that's it about prokaryotic transcription. Further, let's look at eukaryotic transcription now. The transcription in eukaryotes is much more complicated than what we just saw about the prokaryotic transcription, although the steps are the same. This also has initiation, elongation, and the termination phase, but the elements involved are a bit complicated. So in case of prokaryotic transcription, we had just one RNA polymerase, but in this case, we have different RNA polymerases that make different kinds of RNAs. For example, the RNA polymerase 1 makes ribosomal RNA sequences, while the RNA polymerase 2 is involved in the transcription of mRNA, that is the protein coding RNAs, and small nuclear RNAs. And the RNA polymerase 3 is also involved in the 5S ribosomal RNA transcription, and the tRNA, that is the transcription of transfer RNAs. So yeah, so apart from these RNA polymerases, the initiation of the transcription also requires many transcription factor proteins. Also, the transcription initiation needs promoters and upstream regulatory elements like enhancers and silencers. So enhancers and silencers are DNA sequences that regulate the rate of initiation of transcription by the RNA polymerase 2. For example, if an enhancer element is present before the promoter, it enhances the transcription. That is, it increases the rate of transcription of a particular RNA, whereas in the presence of a silencer, the transcription of that RNA is suppressed. So this is just a representation of the promoter region. It consists of the regulatory elements like enhancers and silencers, the transcription start site, and the promoter region, which constitutes of like two conserved sequences called the cat box and the tata box. So the cat box is about 70 to 80 nucleotides in length, whereas the tata box, also called the Hogness box, is 25 to 30 nucleotides long. So yeah, the composition of this region is GGCCAATCT. It has a lot of G's and C's here. Whereas the Tata box, like the name says, it has a lot of T's and A's. So the composition of this is TATAWAW. The W here could be an A or a T. So also the transcription factors or the transcription activating proteins bind to this region and help in the initiation of the transcription. So these were the major differences in the prokaryotic and eukaryotic transcription. And apart from these elements that are responsible during each of the steps of transcription, eukaryotic transcription is followed by some modifications in the new RNA sequence, that is the nascent RNA sequence, to form mature RNA sequences. And these modifications are called post-transcriptional modifications. So yeah, like we just saw, the nascent RNA, also known as the primary transcript, needs to be modified to become functional. So the primary transcripts of mRNA are also called heterogeneous nuclear RNA, that is hnRNA. And these hnRNA, they are larger than mature RNA. So the major processes for the hnRNA to become a mature functional RNA are the following. So one is splicing, and the addition of a cap at the 5' end of the transcript, and addition of a poly A tail, that is like a nucleotide stretch of A's to the end, that is the 3' end of the RNA, and RNA editing. All of these are involved. All of these happen in the post-transcriptional modifications and are also responsible for several functions, and the misregulation of these also lead to diseases and differences in regulation. So let us look at each of these further. One of the major processes of RNA processing is splicing. So the structural genes are composed of coding and non-coding regions that are alternatively separated. That is, like in the gene, that is the DNA, some regions code for proteins while some don't. And when transcription happens, this whole DNA is converted to an RNA transcript. But all of these are not necessary for for instruction to code for proteins. Only the coding regions are retained in the final mature functional RNA transcript. So these coding and non-coding regions are alternatively separated in the primary transcript. So for example, the alphabets here are the coding regions, and these coding regions are called exons. So we could see we have 1, 2, 3, 4, 5, 6, 7 exons here, and these exons are also separated by 7 introns. Introns are the non-coding regions, and they are denoted by numbers here. So what happens in splicing is the non-coding regions, that is the regions denoted by numbers here, are cut off. They are just cut off, and the final transcript only has the alphabets, that is the coding regions. So also this there's a process called alternate splicing, where in some mature transcript we might have only some of these alphabets, like A, C, D and G, like B, E and F could be missing, and we could have a different transcript where A and B are missing but we have the rest of these regions. And it could also be the case that all of these alphabets are present but they are in different orders. So these determine the transcript diversity, and also helps in protein diversity. So this is the reason why there are many diverse proteins that arise from the same DNA region. So splicing is the mechanism that contributes to the diversity. So looking more detailed into the mechanism of splicing, so this is just a simplified pictorial representation. Like we said, we have exons, the coding regions, which are orange here, and longer intronic regions that are non-coding. So what happens is there's a complex of enzymes called that bind together, and this complex is called a spliceosome. It's also called it's called a spliceosome. So what happens is the spliceosome binds to the intronic the junction, the intron exon junction over here, and it just holds the intronic sequences together, and it cuts off the intronic sequence. So the complex here is called lariat. So what happens here is the intronic regions along with the spliceosome is cut off, and we only have the exonic sequences that are retained in the final functional mRNA transcript. So this is the overall mechanism of splicing. So another crucial step is 5' capping. So most eukaryotic mRNAs have a 5' cap. So what is this 5' cap? It is a residue of 7 methyl guanosine, that is methylated G nucleotides, linked to the 5' terminal residue of the transcript. So this the methyl group is attached to the guanosine by a triphosphate linkage bond. So the formation of this cap requires 3 steps. So the first one is the 5' terminal phosphate group is removed by phosphatase enzyme, leaving diphosphate. And then, so we know that like in a nucleotide, one end is like a phosphate group and another end is like the OH group, the hydroxyl group. So in the 5' end we have the phosphate group, and in the 3' end we have the hydroxyl group. So in capping, the phosphate group in the 5' terminal is cleaved off by a phosphatase enzyme. And then the second step is where GTP, guanosine triphosphate, is added by releasing pyrophosphate. So the third step is where the 7th guanine is methylated by methyltransferase enzyme. So methyl group donor is S-adenosyl methionine. So these are just complex chemical terms over here that you need to remember. So here's a pictorial representation of 5' capping. So here is the RNA molecule, and it has a 3' hydroxyl group and a triphosphate group in the 5' end. So what happens is one of this phosphate molecule is cleaved off by the phosphatase enzyme, and we have 2 phosphate molecules. And then what happens is a methylated guanosine along with a phosphate forms a bond with this diphosphate group, and it forms a triphosphate linkage, and so this acts as the 5' cap. So this whole structure is a 5' capped mRNA. So the 5' capping along with the polyadenylation in the 3' end contributes to the stability of the mRNA. So for example, mRNAs which do not have a 5' cap or a poly A tail are very less stable than the RNA molecules that have a 5' cap and a poly A tail. So another step involved in the post-transcriptional modification process of the primary transcripts is polyadenylation. So like we just said, at the 3' end, most eukaryotic mRNAs have a string of about 40 to 20 A nucleotides, that is adenine residues, and this makes up the poly A tail. Like the name suggests, it's just multiple A nucleotides. So hence the name poly A tail. So these adenine nucleotides are added by the enzyme called adenylate transferase. So there's also another kind of modification that happens to the RNA molecules, and this process is called RNA editing. So any type of change in the RNA transcript sequence, like without RNA splicing, like before undergoing splicing, is called RNA editing. So this happens. So this is done to get large diversity of proteins with less number of genes. So editing can happen either by insertion, deletion, or modification to one or more bases of the primary transcript. So this RNA editing was first discovered in mitochondria in trypanosomes. So there are 2 major types of RNA editing. So one is the site-specific deamination of cytosine and adenine, and the other type is guide RNA mediated site-specific insertion and deletion of uridine base, that is the U base. So these are the major types of RNA editing. Let's now look about the first type of RNA editing, which is the site-specific deamination of cytosine and adenine. So the first example is the deamination of cytosine. So like the name suggests, deamination is the removal of an amine group of the cytidine of the cytosine base, and this forms a uracil. So this specific process happens in only specific tissues, and it's also site specific. So this leads to the diversity of proteins. For example, in the intestine, the process of cytidine deamination happens. So in the liver, we have the primary unedited mRNA. So the unedited mRNA where the C nucleotides are not edited, there's no deamination happening. So in that case, this mRNA is translated to a protein called ApoB100. ApoB100. So these genes produces proteins that are involved in the transport of cholesterol in the blood. So whereas in case of deamination, in case of RNA editing, where the deamination of cytosine happens to form uracil, what happens is we get a premature stop codon. So in this case, we don't get the ApoB100 protein. We have the ApoB48 proteins. So these proteins are involved in the absorption of lipids from the intestine. So this is an example of how a modification in one base in a specific tissue can lead to the generation of different proteins and how they are functionally distinct. The other modification is the deamination of adenine to inosine. So here the deamination of the A nucleotide leads to the formation of I, which is inosine. So this usually happens only in secondary structure regions of the RNA. So what happens here is, in case of deamination, we'll have an I nucleotide instead of A. So A is complementary to the T nucleotide, and so when there's an I nucleotide in place of A, what happens is I could be complementary to any of the base pairs. So it could form a bond with G, C, T, or even A. So this affects the secondary structure of the RNA molecules, and it hence affects the functionality of it as well. So the set of proteins that are required for the deamination of adenosine are called ADAR or dsRNA dependent deaminase. So the next type of modification is guided RNA mediated site specific insertion and deletion of uridine base. So guide RNA, it's a part of degraded introns, and it's a small fraction of RNA sequences that just came out of splicing of RNA. So these could act as guide RNA molecules. So these guide the process of RNA editing eukaryotes, and that's why it's called guide RNA. So this is the guide RNA in the biological systems, whereas there are several technologies where guide RNA, like in CRISPR, guide RNAs are synthesized to target specific regions. So that's a different thing. But yeah, it's it does the same function, except that this is natural in the biological system, whereas in CRISPR, the guide RNA could be synthesized. So the steps in this guided RNA mediated site specific modification there are 4 major steps. So the first is the hybridization of the guide RNA with the RNA molecule, and then so this leads to the cleavage of the RNA with endonuclease enzymes, and then so activity of 2-tase, which is an enzyme, and so this leads to the addition of the uridine base in the site where the RNA molecule was cleaved, and then after the addition of the U base, the molecule is ligated using ligase enzymes. So it's just that new bases are incorporated here. So this is a simple representation of this. So in this case, an existing U nucleotides are removed, and here non-existing U nucleotides are added to the RNA molecule. So this could affect various things like the protein translation as well, and could lead to the diversity of proteins. So why is RNA editing important and how it's significant? So it's essential in regulating gene expression of an organism, and RNA editing mutant was reported with strong defects in organelle development. So like we said, the modification of the nucleotide sequence could give rise to mutations or mutant proteins, and this could affect the functions of the cells and hence the organism as a whole. So it is basically a mechanism, like we saw, it also helps increase the diversity of proteins. So this is why modifications are significant or important in this case. So now coming back to our question, which was 5' capping of mRNA transcripts in eukaryotes involves the following events. So they're asking the order in which the 5' capping happens. So if you could recall, we saw a slide about 5' capping, which is one of the post-transcriptional modification steps. So this is one of the steps that happens to the RNA transcript after it is being transcribed. So if you just recall that, the order in which 5' capping happens is, so the first would be the removal of the phosphate of the triphosphate on the first base at the 5' end. And then the next step would be addition of GMP on the 5' end, and then a 5 to 5' linkage between GMP and the first base at the 5' end is formed, and then a methyl group is added to the N7 position of guanine. So, rearranging these, the correct order would be Q, P, R, and S. So, this answers the question. Thank you.